Hi kids, welcome back to the foundation courses with us here at Yupp Master. In our last lecture, we have begun with the whole chapter of cell and its structure and its functions, right? If you remember, we began with the cell organelles of eukaryotic cells, which would include both plants as well as animals too. So we're going to be studying the different organelles of plant cells as well as of animals as well. Now, in our previous video, if you have seen, we have learnt all about the cell wall. We also went ahead, we learnt about the plasma membrane. If you remember, we had called it as a fluid mosaic model. And we also learnt about the cytoplasm. I gave you a brief introduction to what the mitochondria was all about. Now, let's give a brief insight and let's get in details of what is the mitochondria structure looking like, and also why exactly is this mitochondria called as the powerhouse of the cell. Alright, so let's begin. First of all, mitos means thread and chondros means granules. Alright, so if you see in this structure, very clearly you can see in the internal part of it. Can you see how a cross section is taken here? Okay, so there is a cross section basically taken here, and in this cross section it's mentioned how there are thread-like structures and then there are small granules as well. Okay, so these are those small granules present along with those thread-like structures. Now, when we talk about the mitochondria, it's called as the powerhouse of the cell. Let us see why it is called. Okay, so first of all, just a minute. Alright, so we see that in each cell there are around 50 to 50,000 mitochondria present in each cell. Okay, they are rod-like structures, and these rod-like structures are the ones which are actually going to be generating the energy present. Alright, so these mitochondria are present, they are rod shaped, they are small granular structures. Also, if you see here, the mitochondria, they have got two membranes. Alright, which are those membranes? There is an outer membrane which you can see in the dark brown color, and then there is an inner membrane which you can see in the light brown or in the creamish color. Okay, now one more thing you need to know, that the outer membrane that's there, that membrane is going to be smooth and it is around 60 Armstrongs in the thickness, and it is a membrane which is absolutely freely permeable membrane. Okay, whereas when we see the inner membrane, this inner membrane is going to be approximately the same diameter, okay, but it is a membrane which is selectively permeable. So, outer membrane being freely permeable allows everything to pass through it, but then the inner membrane being selectively permeable is only going to allow certain substances to pass through it. Alright, also we see here that it is a double membrane, and this mitochondria, the inner membrane that's there is a fluid inside it, and that fluid inside that inner membrane is called as the mitochondrial matrix. What is it called as? It's called as mitochondrial matrix. So this fluid that you're seeing inside here, all of this fluid can be called as the mitochondrial matrix. Okay, alright. Now we see here that this inner membrane, can you see how that membrane is actually thrown into so many foldings? Can you see these foldings? So it is this inner membrane which is thrown into these infolds. Okay, and when it is thrown into these infolds in here, this is going to help in increasing the surface area. Now, why do we need to increase the surface area there? Well, there are small structures in the inner membrane of the mitochondria, and in order to accommodate more of those structures, why do we need those structures? Because we will see it is those structures which are actually going to be generating the energy. So, however many more of those structures are present, that much more energy can be produced. So, in order to accommodate more of those structures, that is why the inner membrane is actually thrown into folds. Okay. Now, we see that those folds which are present, each fold is called as a cristae. What is it called as? Each fold is known as a cristae. Okay. Now, these cristae have those particles attached to it. Remember, I told you about the particles. Those particles can be called as either oxysomes, or they may be called as F1 particles, or they may be also called as elementary particles. Okay, so basically it is those particles which are going to be on the inner membrane which is going to be producing the energy that it is making, And what are those particles called as? They can be called as oxysomes, or they may be called as F1 particles, or they may also be called as elementary particles. All right. Okay, now we see that there are around 1 lakh to even all the way up to 10 lakh of those oxysomes in each mitochondria. Now, the next question will be, how exactly do these oxysomes or those F1 particles, how do they look like? Okay, so let's see that. Look at this structure here. Can you see this structure here? Let's give it a break. All right, so we see that these oxysomes, let's see how the structure looks like. Look at this structure here. Okay, can you see this is the structure of a mitochondria? All right, let's draw that for you right here on the screen. Okay, so if at all we have this mitochondria here, if this is the inner membrane, then on top of it there is one more membrane which we will call as the outer membrane. Okay, remember, outer membrane is freely permeable, inner membrane is selectively permeable. All right, and then we see that this inner membrane is actually thrown into folds. So this inner membrane can be drawn as folds inside it. All right, so these are infolds seen. All right, now on these folds we see that there are these small units, and we will call this unit as an oxysome. Okay, it is called as an oxysome. Now look at this oxysomes here. So basically this whole outgrowth that you are seeing is the crista, and on top of the cristae you can see the presence of these oxysomes. So this is the oxysome present. All right, the oxysome has a head region, then it has a pedicel region, and then finally it is having a base region. Okay, so this basically is the oxysome. Now we also see that the mitochondria is having its own DNA. Okay, it has a cavity, and the cavity is having a dense fluid. Okay, and that fluid is, we know that is called as a matrix. The matrix has not only DNA, it also has RNA, it also has proteins. Okay, and it also has ribosomes. So in the matrix of the mitochondria, there is DNA, there is RNA, there are proteins, and there are ribosomes too. All right. This is also seen in the matrix of chloroplast. All right. In matrices of chloroplast, also we can see this DNA present. All right. Now mitochondria is called as the powerhouse of the cell because it contains so many of those oxysomes, and those oxysomes are actually the ones which are generating the ATP. ATP, what is ATP? Adenosine triphosphate. And when is this created? This is all being done in the mitochondria during the process of cellular respiration. Okay, so during process of cellular respiration, ATP is created in those particles. What were those particles called as? Either you can call it as oxysome, or you may call it as F1 particles. Okay, so this is the main reason why we are going to be labeling the mitochondria as the powerhouse of the cell. Okay, now coming next, when we see other structures, other cell organelles which are present both in the plant cell as well as in the animal cell, let's move on to another important cell organelle which is known as the endoplasmic reticulum. All right, so now let's just see here. Alright, so let's begin here now. When we talk about the endoplasmic reticulum, understand one thing, that this endoplasmic reticulum, it is absent in the previous cells that we had seen. We had seen that there are cells which are called as prokaryotic cells, like that of a bacteria. So endoplasmic reticulum is absolutely absent there. Okay, and also it is not present in certain cells of our body too. Which are those cells? Cells like the human ovum, cells like mature RBCs, okay, all of these cells do not have endoplasmic reticulum. Tell me one thing, why do you think that RBCs, especially the mature RBCs, they don't have endoplasmic reticulum? Tell me, what is the function of endoplasmic reticulum? Yes, or rather, what is the function of an RBC? Yes, RBC does the function of providing oxygen to each and every part of the body, doesn't it? So if it is providing oxygen everywhere, then naturally that RBC needs a lot of space to hold on to maximum number of oxygen molecules. And who holds on to the oxygen? Hemoglobin. So however much more of hemoglobin is present in the RBC, that much more oxygen it will be able to hold. Alright. So we see here that mature RBCs, if they have endoplasmic reticulum, that will reduce the space for hemoglobin. And that's why mature RBC is not only endoplasmic reticulum, but do you know they don't even have nucleus, alright. So we see that this endoplasmic reticulum absent in prokaryotes, it is present in all the eukaryotic cells, okay. Now we also see that here there could be two types of endoplasmic reticulums. It may either be rough or it may be smooth, okay. And that rough and smooth is based on the presence and absence of ribosomes. So basically, if ribosomes are present, like look at this diagram, this example over here, okay. Over here, if ribosomes are present, then we will label this as rough endoplasmic reticulum because it would be bumpy due to the presence of ribosomes. But look here, this is the smooth endoplasmic reticulum where there are absolutely no ribosomes present, alright. That's why we say it is smooth. Now, this is a continuous system, okay. So now let's see where this starts and where it ends. The endoplasmic reticulum is going to be beginning. Where is it beginning? It is going to be beginning at the level of the nuclear membrane. Can you see that region over there? That is where the endoplasmic reticulum starts. And look at how it is going to be continuous. Can you see how it is continuously there, and it is going all the way up to the plasma membrane. So can I say that this endoplasmic reticulum is a continuous system and it is going to connect one end with the nuclear membrane and the other end with the plasma membrane? Do you understand that? So it is basically a whole bridge between nuclear membrane and plasma membrane. So can I say that it is actually forming the whole skeleton of that cell? Look at how much space it is occupying in the cell, right? So we see it also helps in maintaining the position of other cell organelles too. Like the other cell organelles, look at that around it, look at the Golgi bodies, look at the mitochondria. You can see how everything is going to be in a particular place because the whole cell is held together with that endoplasmic reticulum. So we may also say that this endoplasmic reticulum is forming the intracellular. Intracellular means the part inside the cell. Intracellular supporting framework. Alright. Now we also see that this endoplasmic reticulum can be in three different shapes. Alright. Naturally, it is going to be filled with matrix. Look at the different shapes. The first shape are like that in the tubes here. These are called as cisternae. Alright. Second shape are round vesicle like shape. So these are called as vesicles. Okay. The third one, can you see here how they are branched? Those are called as tubules. Okay. So you have cisternae, you have vesicles, and then you have tubules. Basically, whatever shape may be of the endoplasmic reticulum, internally it is always going to be filled with matrix. Don't forget that, okay. It will always be filled with matrix. So that is the reason why basically nothing is ever left empty. It is always going to be filled with a fluid. Also, one very important thing, that here glycogen, that is stored glucose, and lipids both are synthesized in this and they are not only synthesized, they are also stored in the endoplasmic reticulum. So we can see how smaller substances will reach the cell, and in the cell they are going to build up, and that's why the glucose molecules will come together, they'll be forming glycogen. Fatty acids and glycerol will come together, they will form lipids, and those are going to be stored in that endoplasmic reticulum for further use. Alright. Now, as we said a little while back, that depending on the presence or absence of ribosomes, you can have the endoplasmic reticulum to be either of a rough type or a smooth type. Alright. So, what is the difference basically between smooth and rough endoplasmic reticulum? Well, rough endoplasmic reticulum have ribosomes on them. So, the main function of them would be for protein synthesis. Whereas, smooth endoplasmic reticulum, this is present maximum in the liver, and it is this cell organelle which is helping the liver to detoxify everything. You know that the liver is behaving like a dumping ground of our body. Whatever toxins are present throughout the body, they will first be taken to the liver, and the liver is expected to detoxify all the toxins there. Okay. Also, rough endoplasmic reticulum, as I told you, there are ribosomes, and smooth, all the harmful substances that are taken there to the smooth endoplasmic reticulum are converted to harmless substances, and then ultimately the cell can excrete it out. Alright. Alright, so we continue now with other parts. Okay, first of all, remember that endoplasmic reticulum is going to be continuous with other parts too. Which are those other parts? It all begins with the nuclear membrane. Okay, that nuclear membrane is going to give rise to the endoplasmic reticulum. The endoplasmic reticulum is going to give rise to the Golgi complex. Golgi complex is ahead going to give rise to lysosomes. We will study what those structures are to give rise to lysosomes. Then those lysosomes ultimately are also going to be called as suicidal bags, and those are the ones that would just you can say that they will degenerate or pop out into the cell. Alright. So this is a whole chain of the cell organelles where one gives rise to another, and we will study them in that chain only. So, we have completed with the endoplasmic reticulum. Let's move on and we head on towards the Golgi complex. Alright. Now, this Golgi complex was discovered or described by Camillo Golgi, and Camillo Golgi found this Golgi apparatus or Golgi complex first in the neuron, in the nervous tissue of our body. Okay. So, it was first found in the nervous tissue. What is it composed of? It's composed of these stacks. Okay, so can you see how these are stacks? And these stacks that you're seeing here are, or they can also be called as cisternae. Okay, the stacks may also be called as cisternae. They are membrane bound. What do you mean by membrane bound? Can you see how I've drawn this red line? They have a membrane around them. So those are membrane bound cell organelles. Okay. Now, between 2 to 8 cisternae are usually present in one stack of the Golgi complex, 2 to 8 of them. But look at the count in the liver. Remember, in the liver, the main function is detoxification. So, naturally, all of this will be helping in that and playing an equal contribution in that. So, in the liver, look at how the count greatly increases from 2 to 8 in a normal cell to all the way up to 50 in the liver. Okay. Also, we see what is the function of the Golgi complex. It is concerned with the secretion of glycoprotein and glycolipids. Now, glycoprotein means a carbohydrate along with a protein. Okay. Glycolipid means a carbohydrate along with a fat or lipid. Also, that is the secretion of glycoprotein and glycolipid. Next, storage of carbohydrates, proteins and lipids. Okay, so it is also concerned with the storage of them. That was Golgi complex. So basically the function of it, it is going to take the simple carbohydrates or simple sugars, and it is going to be able to convert those into more complex forms. Okay, so it synthesizes complex carbohydrates from simple sugars. Alright. Next, we can move on towards the whole chain of events. Remember, it all started with, what did it start with? It started with the nuclear membrane. Okay, but nuclear membrane we can study when we talk all about the nucleus. All right, after nuclear membrane, what was the next structure in that chain? Endoplasmic reticulum. After that came the Golgi complex, and finally would come the lysosomes. Now remember, this is lysosome, alright. These lysosomes are also called as the suicidal bags. Now let us just see what the structure is, and let's see why are they called as suicidal bags. Okay, lysosomes are, remember, they are single membrane bound structures. As we saw mitochondria, they were double membranes. Okay, over here when we talk about lysosomes, there's only going to be one membrane. Okay, a single membrane bound structure found in animal cells. What does it contain? It contains an acid which is called as acid hydrolase. What is it called as? It's called as acid hydrolase. And these are, since you have the name ASE, that means that this is going to be an enzyme. And acid hydrolase is that enzyme which is going to be helpful to break down any materials present inside or outside the cell. Okay, so any materials which are present will be broken down. Now because those materials are going to be broken down, lysosomes are going to have a very important role in destroying something which is not necessary. Why? Which enzyme does it have? It has the enzyme called as acid hydrolase. Okay. Now we see that the lysosomes can be polymorphic, means that it can be having different types, different shapes. And where are they exactly arising from? Remember, lysosomes, they arise from the Golgi apparatus. Alright, so from endoplasmic reticulum would arise Golgi bodies. From Golgi bodies would arise the lysosomes. Alright, now coming to the main important part. What are the functions of the lysosomes and why are they called as the suicidal bags? Okay, now we see that this lysosomes is majorly dealing with the function of having an intracellular digestion. Now, what is intracellular digestion? This means that if at all inside the cell only, if there is any cell organelle which is abundantly found, which is too much in number, then, and that too it is too much in number which is not required, then these lysosomes can go and they can engulf those extra cell organelles so that they will not be present. So since they are helping inside destruction inside the cell, okay, you can call it as intracellular digestion. Also one thing, if at all that if at all the cell is becoming too mature, it is too old, it is not able to carry off its own metabolic activities properly, then what happens is that acid hydrolase enzyme which is bound inside the membrane will rupture. When the membrane ruptures, then that acid hydrolase enzyme would spread around the cell and ultimately the cell would just start to degenerate, and that's why we say that the cell would be dying on its own. Since the cell can die on its own like this, then that's why we're calling the lysosomes as the suicidal bags. Okay. Then we also see that not only does it function inside the cell, but this lysosome also has functions externally. And which cells are we talking about? Yes, we are speaking about the sperm. Human sperm, when it approaches this human egg, okay, over there on the egg, there is a coating, alright, and that coating which is present on the egg needs to be penetrated or needs to be crossed over by that sperm. So it is the presence of lysosome which is going to be there, okay. That lysosome is going to be present just in order to break down that coating which is present on the ovum, okay. So once that coating is broken down, then the lysosome can secrete the enzyme, and that of the sperm, it will secrete the enzyme, and that enzyme would be able to or enable that sperm to enter into the ovum. Whichever sperm can secrete that enzyme, only that sperm will gain entry into the egg, okay. So that was an extracellular digestion present, And what is the enzyme that sperm is going to secrete? The enzyme is called as hyaluronidase enzyme. Okay, it's called as hyaluronidase. Okay. So children, what all did we learn about over here? We learned about the mitochondria. Okay, in the mitochondria, do you remember how many of the membranes were there? There were two membranes, an outer membrane and inner membrane. Remember the outer membrane, okay, outer okay. So when we talk about the mitochondria, okay, over there, the outer membrane was freely permeable, okay, and the inner membrane was selectively permeable. Alright, remember one thing, that this inner membrane was thrown into, folds, and those folds are going to be called as cristae. What are they called as? They are called as cristae. Remember, the cristae have small particles on them. What are those particles called as? Those particles are called as oxysomes, or they may be called as F1 particles. Okay, and what is the function of those oxysomes or F1 particles? They do the work of ATP production. Okay, so that was the basic structure of the mitochondria. Okay, remember the mitochondria, the inner membrane, inside it is going to be filled with a matrix, and that matrix is going to be called as mitochondrial matrix. Okay, what does this matrix contain? Remember, it contains DNA, it contains RNA, okay, it contains proteins. Alright, all these structures were found over there. Alright, now let's go ahead and we start with, remember the structures of the Golgi bodies. Alright, now we also began with the lysosomes. We studied the functions of lysosomes. What were they? There was not only intracellular digestion, means digestion happening inside the cell, but also there was extracellular digestion. Now, tell me, do you remember what the function of the Golgi body was? Main function of Golgi bodies was that it was taking simple sugars and it was able to convert those into the complex sugars. Okay. Next, talking about the endoplasmic reticulum, we saw that endoplasmic reticulum could be seen in 3 different shapes. Okay, do you remember the shapes of the endoplasmic reticulum? Those shapes were, first, cisternae, okay, then there were tubules, and then there were vacuoles. Okay, so basically these were there, just the shape of them. Even the endoplasmic reticulum is going to be filled with a matrix. Okay, that's why it is called as endoplasmic matrix. Alright, now we also moved on to the lysosomes. We studied the functions and the structure of lysosomes. Let's move on now next to the cytoskeleton of the cell. Okay, now when we say cytoskeleton of the cell, it means that the skeleton of the cell, or the basic structure or the basic framework that's going to be giving a shape and a support to a cell. So we see here that this is the basic region where it is going to be filling in the whole cytoplasm. Okay, this is found also, remember, it is found only in eukaryotic cells. It works for movement and it works also for support. Okay, and it is going to have its action on the muscle also, and it has its action on the skeleton as well. Okay, look at this diagram here. This diagram here is the diagram of an endoskeleton. Okay, now we see that there are 3 parts of the endoskeleton over here. The first part being microfilaments, the second part being microtubules, the third part being intermediate filaments. Okay, so you have microfilaments, microtubules, and intermediate filaments. Alright, now let us see what each part is made up of. Remember one thing, that all of these parts, being the skeleton of the cell, have to be somewhat a little bit stronger than the remaining parts. So these are all going to be basically made up of proteins. Okay, so don't forget that they're going to be made up of proteins. Now, talking about the first part of the cytoskeleton, that part is called as the microfilaments. Okay, now these microfilaments that are there, they are made up of a protein, and that protein is called as actin protein. Okay, so remember, cytoskeleton has to be made up of a protein. Which protein makes up the microfilaments? A protein called as actin protein. Okay, so cytoskeleton, how many parts to it? 3 parts to it. First part was called as microfilament, second part is called as microtubules, and third part is called as intermediate filaments. Okay, talking about the microfilaments, first of all, it's made up of actin protein. The diameter of this, okay, where is this? Let's see here. Can you see this part over here? Okay, looks like a network here. This here are the microfilaments. Okay, as you can see, it's written, it's made up of actin filaments. Its diameter is around 5 to 9 nanometers. And can you see how it is present right under the plasma membrane? If this part over here is the plasma membrane, this is the region present right under it. Okay. Next, it is responsible for cellular movements. What type of cellular movements? Movements like gliding, contraction, and cytokinesis. Okay. So, whenever a cell is moving, okay, like if you know, when we learn more about the locomotion, about the muscle contraction and everything, even there are cells present there, right? So, it is this actin protein over there which is going to be collectively working in all the cells and helping that muscle to contract. So, yes, it is each and every individual cell and all the cells put together which will coordinate and work together and bring about contraction over there. But due to the presence of which protein over there? Due to the presence of a protein which we are going to be calling as actin. Alright. Okay, so that is what actin protein is all about. Next, when we see after this, after the actin, that is after the microfilaments, we come to the next part of the cytoskeleton, and that part is called as the microtubules. Okay, microtubules also naturally it's going to be made up of a protein, but that protein over there is going to be called as tubulin protein. Okay, now tubulin part, the protein of tubulin is divided into 2 subunits. The first one is called as an alpha subunit and the second one is called as a beta subunit. Okay, so 2 subunits of this microtubules, the protein is called as tubulin, alpha and a beta part. Okay, now we see here that one end of each, okay, one end of each of this is the microtubule is embedded inside the centrosome. All right, so each part of it is embedded inside the centrosome. Where is the centrosome? Here is that centrosome. Okay, so basically when we talk about the microtubules, can you see this spider like apparatus over here? So this is what it is, and this is the part that we are talking about. Okay, this is that whole part. All right. Now, also we see here that this is the basic reason why the shape is given of the cell. They also are going to play a very important role when the cell will divide. Now, what is that role? They're going to be forming spindle fibers. What are they forming? They're forming spindle fibers. When? During mitosis. So, during mitosis, these are going to be helping to form spindle fibers. Also, we see that these cytoskeleton or these microtubules basically, they are present in cilia also, they are present in flagella also. What are cilia and flagella? These are structures which many organisms use in order to locomote. And for locomotion, the cells basically, or either for locomotion of that organism, or if it is a cell inside the body and if it has cilia, then the cilia is going to allow substances to move around it. Okay, so flagella helps for the organism to move, but cilia helps for substances around it to move. Okay, so basically the common thing here is that whether it is cilia or whether it is flagella, it is responsible for movement. And if it is responsible for movement, it has to be strong and it has to be made up of a protein. So what is the name of that protein over here? It's called as a tubulin protein, and remember, tubulin protein has got 2 subunits, an alpha subunit and a beta subunit. Okay, so that was the 3 functions of these microtubules, which was the second part of the cytoskeleton. Now, Coming to the third part of the cytoskeleton. Can you see these green colored structures here? Okay. All of these green color structures that you are seeing are the ones which are called as intermediate filaments. What are they called as? They are called as intermediate filaments. Can you see how these intermediate filaments are actually going to fill in inside the whole cytoskeleton, inside the whole cytoplasm, so that the skeleton, the cytoskeleton is actually spread throughout. So we can see that we have seen three areas, three locations of the cytoskeleton. Which were the cytoskeletons? First, microfilaments, second, microtubules, and third are the intermediate filaments. Okay, the first ones that we saw, the microfilaments, was this part here, the outer ring black color that you saw. Second part that we saw, microtubules, was this part here, the part that looks like a spider on top. Third part that we saw were these that are going to fill into the rest of it, and those are going to be called as intermediate filaments. Okay, so these intermediate filaments, let's see what those are all about. Even these are going to be basically made up of a protein. Which protein over here? These are made up of a protein called as keratin protein. Okay, so the first protein that was present was actin protein. The second protein that's going to be present is going to be called as tubulin protein, and the third protein that's going to be present is called as the keratin protein. So three protein names we just learnt over here. One is actin, second is tubulin with two subunits alpha and beta, okay, and the third is called as keratin. What are these all contributing to? Formation of the cytoskeleton. Remember, all of these three are proteins. Okay, all three are proteins. All right. Now we see here that the diameter of these, they are around 10 nanometers, okay, diameter. And what is the main function of it? It's going to be providing a tensile strength, okay. It's going to be helping that cell to resist any stress that's coming on it, any external pressure that comes on it. It helps it to resist it, okay. So three parts here. Remember those names of those three parts. First part was called as microfilament. Second part was called as microtubules. Third part is called as intermediate filaments. Okay. Microfilament was made up of actin protein. Microtubules was made up of tubulin protein alpha as well as a beta subunit. And the intermediate filament is made up of a protein called as keratin protein. Okay. I hope this is clear here right now. So we have done a majority of the cell organelles belonging to the eukaryotic cells. Yes. Which all cell organelles did we cover? We covered, starting with the first one, plasma membrane. Okay. Okay. Then we moved on towards the cell wall. Do you remember another name for plasma membrane? Couldn't we call it as fluid mosaic model? Fluid mosaic model. Second is the cell wall. Alright. Remember, the cell wall is a structure which is present only in plant cells and not found in animal cells. Okay. Then after the cell wall, inside to that was the cytoplasm. Okay. After the cytoplasm, we moved on. We went to a structure. Do you remember the cytoplasm? I told you that it was a colloid structure. Okay. And also I told you that there was a specific type of movement of the cytoplasm. We had given it the name cyclosis. It was also called as the streaming movement. Okay. After cytoplasm, we went on to the mitochondria. Okay. Mitochondria had the inner membrane which had infoldings, and those infoldings were called as cristae. Cristae had small particles on it, and those small particles are called as oxysomes. Okay, or they may be called as F1 particles. Okay, oxysomes or F1 particles. After this, after the mitochondria, we headed on next towards the endoplasmic reticulum. moved on towards endoplasmic reticulum, and this endoplasmic reticulum was the whole framework of the cell. Do you remember that the endoplasmic reticulum was a chain which was connecting the nuclear membrane connecting it with the plasma membrane. Okay, it is connecting the nuclear membrane with the plasma membrane. After endoplasmic reticulum, next part comes, it was called as the Golgi apparatus or complex. Okay, what was the main function in Golgi apparatus? I need you to remember the function of it. It was doing the work of converting simple sugars to complex, okay, and also it was giving rise to another structure which was called as the lysosomes. Lysosomes were called as suicidal bags. Why? Two most important functions of lysosomes. Which are they? First of all, it contains an enzyme which is called as acid, okay, hydrolase. Alright. The functions would be intracellular digestion and also extracellular digestion. Okay, in intracellular digestion, that's why we are also calling them as suicidal bags. Okay, and extracellular digestion is happening at the level of the sperm and egg for fertilization. Okay, so these are the different parts of the cell that we have covered today in today's lecture. We also headed on and we completed the lecture with cytoskeleton. Remember the three parts: microfilaments, microtubules and intermediate filaments. Okay, in the next lecture we'll be heading on towards the parts of the nucleus. So stay with us, stay tuned with us at Yupp Master with the NTSE and the foundation courses, and pretty soon we'll be discussing about the MCQs that will come of this chapter itself. Alright, so